Hello and welcome to Chapter 8, Part 1 for EFB103. This is all about photosynthesis. Let's get started. Now we can split the kinds of feeding of organisms into these categories, autotrophs and heterotrophs. So plants are autotrophs. What that means is they're self-feeding, that's autotroph. Heterotrophs eat different things. And we're heterotrophs. These beetles are heterotrophs. But you can see that heterotrophs rely ultimately on autotrophs. Now they may be directly connected, like these beetles feeding on this plant, or when we're eating a nice salad, or maybe indirectly connected, where heterotrophs are eating other heterotrophs that had eaten plants before them. All right, so there's all kinds of these autotrophic organisms, and we're really focusing in on the photoautotrophs. So what that means specifically is things like plants use light to build that food and self-feed. There's that autotrophy. All right, so this is all about how plants do that. Notice here there's other examples. So unicellular protists can do it, algae, different kinds of prokaryotes are photoautotrophs as well, but we're going to really mostly be focusing on the plants with some diversions here and there into these other areas. Okay, question. The biomass, or dry weight specifically, of a tree, or most of it, comes from what? Okay, yeah, so given your options there, air is the answer. Now, it's kind of a vague term, air, but it's specifically the carbon dioxide in the air. That's what's important, and that's what we're going to see is what's used and how plants build sugars, and then those sugars form cellulose strings, and ultimately that's what makes up most of a plant. All right, so let's zoom in and into the structure of a plant. We're going to take a cross-section of this leaf here, and we can see there's the outside area of the leaf. There are these pores in the surface of the leaf. They're called stomata. Stoma basically means mouth. And that's where gases, carbon dioxide, oxygen come in and out. And here is a mesophyll cell. So we'll just zoom in on that. And it contains chloroplasts. So that's what you see, these dark green dots here. So you got to think back to your chloroplast anatomy from a previous chapter. Here's a quick reminder what chloroplasts look like. This is where the action's going to be happening. So chloroplasts have that double outer membrane. What's really important here with photosynthesis are these stacks of thylakoids. So these membranous structures, this provides additional compartmentalization within the chloroplast. A stack itself is called a granum. And then within that is the thylakoid space. That's also going to be important. So make sure you're good on your chloroplast anatomy. So let's do the 10,000 foot view of what's happening in photosynthesis. So light energy is harvested by protein complexes in the thylakoids. You need water for this to happen as well, photosynthesis to happen, not that light absorption per se. When that light energy boosts electrons to a higher energy state, those can be passed off to this NADP plus, and then it becomes NADPH. That's an electron carrier. We're going to make a bit of ATP. We'll see how that works. And we're going to produce oxygen as a consequence. So the oxygen that's produced by plants, this is coming out of the light reactions here. All right, so we've got a bunch of ATP and reducing power down here from our electron carrier and our ATP. What is that used for? Building sugars. And that feeds into this cycle called the Calvin cycle. This is where that carbon dioxide gets used. So, plants are made of air. They're pulling that carbon dioxide out of the air and using it to build sugars. And they're using the energy of ATP and this reducing power, the high energy electrons that are carried by this NADPH. Now, this is a good time to look at photosynthesis just as a whole, and where the oxygen comes from that's being produced, and where that carbon dioxide ends up. We'll see what happens there. But you can think of this equation. It looks very similar to cell respiration in just flipped around. But it's not. This isn't just cell respiration running in reverse. The formula looks similar, but obviously just some different things are happening. But we can march through this and see where those oxygen atoms come from. So we could have a couple of questions. Does the oxygen come from, say, the carbon dioxide that's feeding in, or does it come from water? And so you could set up an experiment here, like I'm showing you here in the bottom. You can use an isotopic oxygen, oxygen 18. And so here we're going to use isotopic water. So it has the heavy oxygen. And then in experiment 2, we're going to use our carbon dioxide, and it has the heavy oxygen. And ultimately, we want to see, well, where does it end up? So if we use the heavy oxygen here in H2O, this is where the gas oxygen is released at the end. So when sometimes students think, well, I put carbon dioxide in, oxygen comes out, there you go. There's oxygen here, there's oxygen there. That's not where it comes from. The oxygen that plants produce comes from water. And you can see that in experiment 2 here. It's like, well, let's put heavy oxygen in CO2. Where does it end up? It ends up in the sugars that we're making, and some in this water here, the water that's produced. All right, so here we can look a little more simply, maybe. Here we see our reactants and our products, and you can see where each of the atoms go that feeds into photosynthesis, and then what comes out. Now, I will point out that this is showing how to make a whole glucose molecule from 6 carbon dioxides, but ultimately, if I go back, this is a point here. The direct product of that Calvin cycle that builds the sugars, it's not glucose, but it's actually a 3-carbon molecule, and 2 of those can be used to make glucose. We'll see that in more detail when we get into that. All right, so let's look again, the whole view of this. Water is oxidized. That's what then ultimately oxygen is produced from that. Carbon dioxide becomes reduced. Its reduction is gain. It's picking up electrons. Think about how in cell respiration, glucose had electrons ripped out of it, and then ultimately CO2 was produced. To start to build sugars again, you need to put high energy electrons back in. So we need to reduce that CO2, meaning gaining electrons. And that's how we produce that sugar. So the light reactions provide the energy. So you need that energy input. So this is an anabolic reaction. And this can be thought of, again, as looks like the reverse flow of electrons compared to cell respiration. Again, that doesn't mean that these are just reverse processes. Photosynthesis is a very different process, but if you just look at it very simply, that you can apply that. So here was cell respiration. We took high energy electrons and eked out the energy to put into ATP synthesis or proton gradient formation. Ultimately, those protons end up the electrons rather end up being passed to oxygen producing water. And so photosynthesis is taking the electrons out of water and boosting them back up in energy levels, and then plugging them into glucose, or precursors to glucose rather. How does this light absorption work? Well, you can look at the electromagnetic spectrum here, and we see visible light, this narrow slice in the middle here. And this is what powers photosynthesis, visible light. And if you look here, you can see the different wavelengths. Again, this is just a narrow slice here in the nanometer range, 380 to 750 nanometers. And you can see the different colors. So we go from violet to red. And the shorter wavelengths are on the left, and wavelengths get longer as we go to the right. And then there's this relationship between energy as well. And this connects to the idea that light is has properties of waves and also particles. So you might be familiar with this idea that Einstein really developed the modern concept of, is that these photons are discrete elementary particles and packets of energy, and that energy is inversely proportional to the wavelength. So the shorter the wavelength, the higher the energy, the longer or bigger the wavelength, the lower the energy. So how do plants absorb this? They have pigments. So these are large chemicals, I'll show you structure in a moment, that help absorb that light. The primary pigment is chlorophyll A. So this is something you might have heard of. But there's more than chlorophyll A. There's also chlorophyll B. And there are some other accessory pigments called carotenoids that help absorb light in those different wavelengths. Chlorophyll A and B are very similar, but there are slight structural differences that cause them to absorb slightly different wavelengths of light. And so let's have a look at these. So here's chlorophyll. Notice this hydrocarbon tail. This is going to help it stay anchored in a phospholipid bilayer. So this is the part that's going to be within that bilayer. And this part here is what will project out. And notice there's looks like a lot going on, but that's important for being able to absorb that light. Notice you need magnesium as well. So you need that cofactor there. And so here's the difference here between chlorophyll A and B, a methyl group in chlorophyll A and a carbonyl group in chlorophyll B. But what really matters is how these things work. And so you could look at the absorption spectra of these different pigments. So here we have carotenoids, chlorophyll A and B, different lines here, dotted gray or black, and you can see where they absorb. Notice all of them really do absorb pretty well in the blue and violet here to some extent. And think back to that idea of the relationship between energy of those photons and wavelength. Higher energy, shorter wavelength. And ultimately, what is not being absorbed is what's reflected. So you could ask, well, why do plants look green? Let's go back to the absorption spectrum again. What's not being absorbed very well by these pigments? Look, green, yellow, maybe getting to orange a little bit. So that's why plants look green, because that's what's bounced back. That's what's not absorbed. These other colors are absorbed. And so what you see, anything that you see, that's what's being reflected and not absorbed. It's the other colors that are being absorbed. So here we see it again, and we can start to ask the question, okay, well, here's what these pigments absorb, but what is most effective at driving photosynthesis? So, well, I mean, they have good absorption down here in these color ranges, so that seems good. But we can actually measure photosynthesis and see what happens, and then overlay it to these absorption spectra. So here is what's called the action spectrum. So this is looking at rate of photosynthesis, as measured by O2 release, at different wavelengths of light. So again, we see, well, it looks pretty good around in that purple and blue, as we might expect by the absorption. We can jump back here. Here's also absorption out toward the red, and photosynthesis looking pretty good there too. Photosynthesis isn't very effective. There's a bit, but it's not as good in these green and yellow wavelengths. Okay, so question. Here's a chlorophyll A absorption spectrum, just like we've seen. There's that action spectrum, or rate of photosynthesis, in the dotted line. Why are these things different? All right, yeah. So they're different because these are really two different things. The chlorophyll spectrum is the chlorophyll spectrum, the absorption spectrum, but the action spectrum is actual photosynthesis. And that is going to be driven by all those pigments, not just chlorophyll. All right, so let's see what's happening when chlorophyll is absorbing that energy. How does this work? How does this translate into plants making their own food? And so it might help to think of this in a test tube, or well, a flask in this case. You can take chlorophyll out of a plant. It's just chlorophyll in a solution here, and you can see it glowing or fluorescing when you shine light on it. So what's going on there? Well, here's the little cartoon version. Light is shining on the chlorophyll molecules, and what goes on is an electron gets boosted. Remember, think of energy states of electrons and those orbitals. If they go to higher orbitals, they have more energy. And so what's happening is they're absorbing the electrons, get energy shunted to them, and a special pair of electrons will be basically kicked out of chlorophyll. So they get so excited, they jump out of chlorophyll. And then in the tube, in the flask example here, they have nowhere to go. So they drop back down. That's this other arrow here, thunk, back down into chlorophyll, down to their ground state. But in that, you've gone from something with potential energy, this high energy electron, back down to its ground state. What happens? Heat and light is produced, in this case, fluorescence. And so this is the basics of what's happening here. We're boosting up electrons to higher energy state. Just in a tube, they'll just go back down, produce heat and light. But life can capture that energy of these high energy electrons and make them do work, just like we saw in cell respiration. So let's zoom in on this now. So here's our chloroplast. Here's our thylakoid membrane where all the action's happening. Here's chlorophyll molecules embedded in this protein complex. What are these protein complexes? They're called photosystems. And so here's a zoomed in version of this. Is a 3D, well, it's not 3D, but you can kind of imagine it in 3 dimensions here. The purple stuff is protein, and then embedded within that, in the thylakoid membrane, are chlorophyll molecules and other accessory pigments, but we're highlighting the chlorophyll here. Here's a more cartoonish version of it, but a little easier to digest. You've got this photosystem complex, and you have pigments in what's called the antenna complex. That's the sort of region on the outer part of this photosystem. And that might contain different kinds of chlorophyll and other accessory pigments. And we see this energy from light being absorbed by these pigments, and that energy kind of bouncing around and ultimately being funneled to this reaction center area. Now, it's important to remember these black arrows, these are not electrons being passed around. That's something that some students have a misconception about. That hasn't happened yet. It's just energy. It's not enough energy to kick any electrons out of anything yet, but it all gets funneled to this one place, and it's all happening very dynamically until there's enough to kick an electron out of this special pair of chlorophylls in the reaction center. This is just another way of looking at it. See energy bouncing around. Ultimately, an electron gets kicked out of this, out of those chlorophyll A's. Now, this is also called the photoelectric effect. So if you've ever had a plasma screen television, this works on the same principle, that electrons are being boosted and then they drop back down, and that emits light. That's the photoelectric effect. Again, living things like plants, they're going to kick those electrons out, but they're not going to just waste them by having them drop back down and produce light and heat. We're going to harvest that energy along the way. All right, so what's the primary function of pigment molecules in that light harvesting complex of the photosystem? All right, so the answer here was that they are basically a funnel to channel that energy down into the reaction center, where it will collect and build up and kick those electrons out. So let's look at these photosystem complexes and how they work together, and how we really now are getting into the light reactions of photosynthesis. So this photosystem 2 is often considered to be acting first in this process. Now, keep in mind, this is all very dynamic, but we're going to go through this step by step. So photosystem 2, here in the middle of in the reaction center, is this light absorbing complex at wavelength 680. So this is called the P680 complex. And so when energy is harvested by these accessory pigments, they're passing the energy to that P680. Ultimately, that's where the electrons get kicked out from. So they get higher and higher energy states, and then finally, boom, they're gone. Now this photosystem complex is down a couple of electrons, right? So where do those come from? Where do they get replaced by? It's from water. So it's the splitting of water, which, if you remember, these are kind of low energy at this point, right? Think back to cell respiration. That oxygen finally was past those low energy electrons. So photosynthesis is taking them, and it needs to be pretty strong at taking electrons out of this very stable compound, which is water. And so this is considered one of the strongest oxidizing agents in any living thing, this P680. And it will take the electrons from water, then producing oxygen and hydrogen. So now this has got low energy electrons, which again absorb light. They can be boosted up again. Okay, so photosystem 1. This is the second photosystem in our sequence here, but it's called PS1 because it was discovered first. Simple as that. And it absorbs its chlorophyll, its reaction center chlorophyll, tends to absorb wavelengths of light better around 700 nanometers. And so that reaction center chlorophyll is called a P700. Now, both these photosystems are pretty similar. The subtle difference is, well, where they occur in this sequence and their light absorbing properties, 680 nanometers for PS2, 700 for PS1. And of course, it is really important what's happening in the middle here, and we will get to that. But let's look at this electron flow. So here we're seeing electron flow with the orange arrows, ultimately being passed off to this electron carrier over here. But let's look at this in some more detail. So electron flow can be cyclic, so cyclic electron flow, and we're still going to be able to produce some ATP as a result of cyclic electron flow. And this is called cyclic photophosphorylation, which sounds, again, like a lot, but again, it's cyclic. The photophosphorylation, all this means is we're building ATP using light energy. Photo, light, phosphorylation. Phosphorylation of what? ADP to make ATP. So again, looks like a big word, but if you break it down, it's pretty simple. Non-cyclic electron flow. This is what's happening in plants most of the time, and we'll see how that works in a second. Let's start with this idea of cyclic flow. And I'll use this flask example again. So we're not making ATP here. So we're not really phosphorylating anything in this lab example, but the idea of it being cyclical works. So many photons excite those electrons, boom, they go to an excited state, drop back down, heat and light produced, and that's the cyclic flow. The electrons end up back in that same chlorophyll complex or a neighboring one. So that's cyclic. We see this in prokaryotes, for example. So a single photosystem complex, electron gets boosted up to high energy. It is passed, like we saw with electron transport in cell respiration, through a set of redox reactions, through this pump, which pumps protons. It's not directly producing ATP, but it's pumping protons, which creates a gradient. Guess what? That produces ATP, just like we saw in cell respiration. So we see this in prokaryotes commonly. So you may be wondering, well, prokaryotes don't have chloroplasts, right? Because they're prokaryotes, they don't have organelles. But remember, the chloroplast itself is derived from an endosymbiotic event of a cyanobacterium into a eukaryote, and that's where chloroplasts come from. So these structures are found in prokaryotes. Usually that they're going to be found in the cell membrane. So they some prokaryotes have an inner and outer cell membrane. So this would be the inner cell membrane. There's a space between that and the cell wall. That acts as a place to pump protons. And some prokaryotes actually do have this compartmentalization, these thylakoid-like membranes that creates that compartmentalization where, again, protons can be pumped, creates a gradient, and then produce ATP. All right, how about non-cyclic photophosphorylation? Now, this occurs in plants, eukaryotes. This also occurs in prokaryotes. Again, cyanobacteria, right? Where did chloroplasts come from? Cyanobacteria. So some prokaryotes do this as well, not just cyclic photophosphorylation. And we can follow the electrons here. So energy absorbed from light boosts up the energy levels of those electrons. Now they're at a higher energy state. See free energy over here on the left. They're passed through a set of redox reactions through a proton pump. That's going to pump protons into the thylakoid space, creating a proton gradient. It just comes up so many times. If you don't have the concept of proton gradient at this point, write it down. Make sure you understand it. Go back to cell respiration. But on we go. Pumping protons, then passing electrons to photosystem 1. Now there's got to be room for those electrons to be passed. So meanwhile, photosystem 1 has been collecting energy and kicking its electrons out, up, again, higher energy levels, pass through redox reactions. And then these ones coming in over here can now fill that void where those electrons have left and gone up here. Where do they go? Why is this non-cyclic? They don't end up back in the photosystems, but they're passed to NADP plus by this enzyme, NADP reductase, creating NADPH, which is the electron carrier. And it's going to carry those off to the Calvin cycle, which is going to build our carbohydrates. So let's talk a little bit about NADP plus or NADPH. It looks a lot like good old NAD. The difference is this P, a phosphate. And so there it is on the end of this sugar. Now, it can be helpful. I often think of this as NAD plant. So thinking photosynthesis, or could be NAD photosynthesis. Maybe that's better. But it's NADP because it has this phosphate. And so these guys are your electron carriers in photosynthesis and cell respiration, respectively. All right, so you got to have the electron carrier, right? Or else this is going to get backed up, right? If you can't pass these high energy electrons to NAD+, then these ones, they have nowhere to go. These ones can't be kicked out. These ones can't backfill, and so on. Everything just gets backed up like a traffic jam. And so in those cases, in low NADP plus environments, what do plants do? This is going to grind to a halt their ability to produce ATP in this manner. It's not the end of the world. There is a way for plants to do this. They can revert to cyclic photophosphorylation. So what happens, instead of passing those electrons off to NAD+, they just feed them back into that electron transport chain. And again, they can keep pumping protons. That means they can keep making ATP. And you've cut photosystem 2 out of this process. And that means you're not going to produce any oxygen. Because if we go back, this is where oxygen is produced by the donation of the electrons from H2O, but that's not happening here. So plants have a way to get around these low NADP conditions. Okay, so what's the function of those electron transport proteins in the thylakoid membranes? All right, yes, pumping protons, create a proton gradient, and that potential energy used to make ATP. All right, let's look at this step by step really quickly. We've been over it already, but let's look here. Light, energy absorbed, bounce around to the P60 complex. Electrons get kicked out to a higher energy state to that primary acceptor. Now you have this void. These are down a couple of electrons, right? So they have to come from somewhere. Where do they come from? Water. That's where oxygen is produced and a couple of hydrogens. Meanwhile, these high energy electrons are passed along and through this proton pump which creates a proton gradient. Doesn't directly produce ATP, but it makes a proton gradient. Now, these backfill here because what's been happening in photosystem 1, light energy absorbed, electrons kicked out to a higher energy state, that leaves a void here. You need a couple of electrons to fill this. So in they come from the electron transport chain. Meanwhile, the electrons over here are being passed through a couple of redox reactions to NAD+. All right, we can look at this now a little more spatially. So here's the thylakoid membrane. This green area is the thylakoid space. Out here is the stroma, where all this stuff is happening really on the thylakoid membrane, but you need the thylakoid space to act as a place to create that proton gradient. And I encourage you to check out some of these video links. When you get to the PowerPoint slides, download those and click on these links. That can show in a very animated, dynamic way what's happening here. But here's that proton gradient. Protons being pumped into the thylakoid space. They're going to move down their gradient through ATP synthase. The other thing, too, to notice, it's not just the pumping of protons that contributes to that gradient, but look here. Every time water is split, producing oxygen, it also produces a couple of protons. So those are also contributing to that proton gradient. Here again, just another way to look at it, pumped protons plus these protons, and then you see both are contributing to that proton gradient. And then this is the other thing we're going to connect to shortly. Where did those electrons go? High energy electrons, and some of that energy from the ATP we're making off to build sugars in the Calvin cycle. Just showing a couple of other figures that I think are helpful. If you think about just the flow of electrons here, initially from water, get boosted up, passed along, pump some protons into the next photosystem, and then ultimately into NADPH. And here's another way to look at this. We're now looking at multiple things, not just electron flow, but protons. And then again, the lumen of the thylakoid space is where you see a lot of those protons. And that's emphasized here in this image, just showing that proton gradient. Not a lot in the stroma, lots in the thylakoid space or thylakoid lumen, and so on. Okay, so if you expose plants to a toxin that makes those thylakoid membranes leaky to small polar molecules, what's going to happen to ATP production? Okay, yeah, so if this happens, that means hydrogen can leak across the membrane, and then you're losing that gradient. That means you're losing that potential energy, and ATP production in the chloroplast, at least, is going to decrease. All right, now let's contrast cell respiration and photosynthesis, because we've seen some parallels here, and it's good to connect these ideas, and really you're just building on things that you've already learned. Think of proton gradients, right? So in the mitochondrion in cell respiration, that intermembrane space is where we saw high proton concentrations, and down their gradient go the protons through ATP synthase. The same thing happens in chloroplasts. It's just that the thylakoid space or the thylakoid lumen is where the high proton concentration is taking place. So you can see these parallels, and electron transport chain system is slightly different in both of them, but both are pumping protons, both are a set of redox reactions. And so there are a lot of parallels between these two processes. All right, so with that, we'll find out what happens next in photosynthesis. We've harvested some energy. Now let's build something.